welcome back to At the Bench, the Neonatal Physician Scientist podcast of the Incubator, where we are incubating discoveries. I'm Misty Good and a neonatologist scientist and division chief of neonatology at UNC Chapel Hill. And I have the absolute pleasure of co hosting with Dr. Betsy Crouch. Betsy, do you want to introduce yourself?
Hi, everyone. Happy New Year. I'm Betsy Crouch, and I'm an assistant professor of pediatrics and stem cell biology at the University of California, San Francisco. Also, very happy to be here and happy to get to have a nice discussion with our guest today, Dr. Bunmi Olaloye. She is an assistant professor of pediatrics in the Division of Neonatology at Yale School of Medicine. Bunmi, thank you so much for joining us. Do you want to provide a brief intro to you and some of your passions to get us started today?
Yeah. Hi, Misty and Betsy. Thank you so much for having me on the podcast today. I'm super excited to talk to you about my journey to becoming a neonatologist and physician scientist. As you mentioned, I'm currently a junior faculty at Yale, and I'm transitioning to independence while working with my mentor, Dr. Lisa Konikova, who is an immunologist and neonatologist here in New Haven. And like a lot of physicians, I was interested in a career in medicine pretty early on, inspired by my dad, who was an orthopedic surgeon. Um, but then in high school, I took a little detour and was sort of interested in engineering after an amazing class with a physics teacher, and figured that I would do sort of a biomedical engineering undergraduate degree. It would be a good way to combine my interests in human physiology and physics. Um, and then at that point, got interested in maybe doing an MD PhD, but didn't get into any MD PhD programs. But yeah, I'm excited to sort of talk a little bit more about my journey to becoming a physician scientist who is trying to understand why babies develop necrotizing enterocolitis, and that has sort of sparked an interest in early life immune development, as well as spontaneous intestinal perforation. So thank you again for having me.
Yeah, I'm personally so excited that you're on the podcast and we get to talk about your work, which is really transforming the field and taking a completely unbiased approach to this disease. And so I'm just really thrilled that you're here to chat with us today. Yeah, I was wondering if you could tell our listeners. I know a lot about your background and training, but maybe you could tell us or tell our listeners about your your training and mentoring and through that, how that uh, got you to the stage.
Yeah, thank you for that question. So, like I mentioned, I sort of always knew I wanted to become a physician, and in doing an undergraduate degree in engineering, I sort of was interested in potentially the science of medicine, but like I said, I didn't get into any PhD schools, and I did my medical training at Rutgers, um, New Jersey Medical School in Newark. And while I was there, I had the opportunity to do a sub-I in the NICU during my fourth year, and that really sort of inspired me to want to become a neonatologist. I was able to work with Dr. Fofa, who was an amazing neonatologist who I think is still at Rutgers. Um, and then that and then after being in New Jersey for a few years, I wanted a change of scenery, and so I went to my residency at the University of Texas in Galveston, where I got a really great training in sort of becoming just like a well-rounded we had a lot of exposure to neonatology. And at the end of that, I still sort of felt like there was maybe something that I was interested in that I hadn't pursued, which was science. And so I was lucky enough to do my fellowship at the University of Pittsburgh. And while I was there, I was like, this is my last chance to see if I could have a career in science. And I looked at a few different labs, and I ended up in Dr. Lisa Konikova's lab, like I had mentioned earlier. And I was really sort of inspired to try to study neck, because as we know, a lot of babies just suddenly develop neck. I remember one particular patient during my second year of fellowship was like, I was taking care of this baby for a week or 2, and everything seemed fine, and then sort of out of the blue, the baby got really bad necrotizing and then unfortunately did not survive, and so, in doing a lot of reading and research, there just really weren't a lot of answers. And I was hoping that, I would get into the lab and study NAC and try to identify biomarkers and therapeutic targets to identify babies who would develop NAC, and then potentially identify potential therapies, which I know Misty is also working on. But then that journey has sort of really expanded um, to sort of include trying to learn about how peripheral immunity develops in preterm infants because we don't know that much about it. And then also in our initial study, we had included babies who had spontaneous intestinal perforation as like age matched controls. But it turns out that also has sort of like a distinct immune landscape. And so that second piece has sort of also become um, a passion project as like there are even fewer studies in SIP. So yeah, that's kind of the whole story.
That's awesome.
So many great things to talk about. Furiously taking notes, like, "Oh, we have to talk about sit versus neck. Oh, we have to talk about," I think another theme that I also wanna highlight is, I also didn't get into medical school the first time I applied. I was a senior in college, um, and I think I applied to MD programs, and then when I didn't get in, went to the NIH and did 2 years of research and then got into MD, PhD programs. So I do think and yet here we are, like, in a similar scenario, So it's like, I think that there are many, many routes to, a physician scientist career, and, I think it's about taking the best opportunity that's in front of you.
I agree. I think it's important to highlight that, right? Because, so many of our listeners that may be at various stages in their training. And so don't lose hope. Keep going. And you never know where you'll end up. So I think it's great to highlight that.
Yeah, I mean, I think that I've been fortunate to have great mentors and being in places where there were a lot of great resources existing, and so that's sort of been incredibly helpful on my journey so far. And yeah, I think that to echo what you both said, it's like if at first you don't succeed, just like try again.
Yeah, and you'll learn things, I think, through whatever path is in front of you. Um, yeah, there's a resident right now in my who's working in my lab, and I admire her because she came to talk to me and she said, I want to try some, bench science. And we have her doing like a cool translational project, trying to do some work on a project that we have understanding how there are neurodevelopmental differences in kids with congenital heart disease. So making some brain organoids in kids with IPS, in IPS lines from kids with congenital heart disease. Um, and the reason that she said that she wanted to come be in the lab was because she said it scared her and she wanted practice overcoming the things that were scary to her. And I thought, that's a winning attitude. Yes, please come in the lab,
Yeah. Yeah, that's an amazing attitude to have. I remember sort of when I first joined the Concova lab, like I had done some, engineering related research in undergrad, but I hadn't done any sort of wet lab research. And so, I kind of showed up and I was like, yeah, like, this is what I want to do, but, you know, I am willing to like learn. And, I did some reading and at first I was like, well, maybe we could like look in the urine or like maybe we can look in the blood. And then, Lisa, my mentor was like, well, we have some samples, some intestinal tissue samples, and this lab does like mass cytometry. So, that's where you can start and then I was simultaneously working on a protocol to sort of isolate immune cells from small volumes of blood. And so both of those things have been kind of an opportunity to learn about immunology because um, for most physicians, the immunology we get in medical school is like 1 6-week course, and it's very basic. Like these are B cells, these are T cells. But being in a mucosal immunology lab was sort of an immersive learning experience in a lot of different things, and I'm still learning. So yeah, to kind of everyone out there who's interested in something or has, a clinical question that you're interested in tackling, it's important to sort of like you can see the sort of mentors that are around and look at the resources you have and try to learn as you go.
I think that's great. And, especially not having I mean, that was me too, especially we both trained in Pittsburgh, so I love that connection. That was me. I didn't have any basic science experience and was learning to pipette when I was second-year fellow, so, I was right there with you. I did not, learn mass cytometry, at that time, but certainly, that is a technique that you know, is not new anymore, but was new at the time. I wonder if you could talk a little bit about, learning that, and the different stages, and then how your science and, analysis, all the different techniques, that you utilized, how that's evolved since you were a fellow.
Yeah, yeah. I mean, so like I think that the lab was just like myself, and there was a grad student and like the research tech, so it was really small when I joined. And so I think that um was a great opportunity to get sort of hands-on experience in all the steps that were involved. And so the lab we work had a protocol to collect intestinal tissue samples and bio-bleed, cryopreserve them. And that was from Dr. Konkova's postdoc. And so I learned sort of how we got the samples. And then I was able to work through just sort of the actual steps of performing the experiment. So, like learning to isolate the single cells. And I think in the beginning, I showed up every day and I was eager to learn. And at the time, I don't think I realized one how precious the samples were because you get what one sample and like you don't you don't have an infinite number of samples, or two, sort of like all the potential things that could go wrong. But we were fortunate enough to have a cohort of samples, and for our initial sort of mass cytometry experiment, we obviously were super interested in understanding necrotizing enterocolitis and the biobank of samples that was available in the lab. So for this experiment, we included um, some normal unaffected prenatal samples that we compared to postnatal samples obtained from babies who had surgeries for necrotizing enterocolitis, patients who had surgeries for spontaneous intestinal perforation, and then patients who had surgery for congenital intestinal anomalies like intestinal atresia. And basically, that experiment, the main goal is to first sort of take these samples and isolate the immune cells from the intestine, which we did over a 1 day process. And then the next day, we take our cocktail of antibodies to identify various populations of interest. So, in the immune cells within the intestine, including monocytes, neutrophils, T cells, B cells, natural killer cells, ILCs, and then you get an abundance of data because you have a lot of cells. And so I had the opportunity to learn how to take all of that data and using sort of various pipelines available through um, our studio to kind of make some meaningful analysis from there. First, doing some like traditional FACS type gating to identify which were the immune cells, and then subsequently looking at various populations. And what we were able to see from there was that there's a distinct sort of immune landscape um, in necrotizing enterocolitis that's different from all the other groups that we compared to. And that was characterized by an influx of neutrophils and macrophages. Some of that was known from previous studies, but we were also able to sort of subtype um, the neutrophils, and we saw sort of three distinct populations of neutrophils that we thought corresponded to timing since the injury. And so some neutrophils were kind of more, we thought were early, early neutrophils, and then other ones were sort of like late, more age neutrophils, and then some neutrophil DC like hybrids that the next samples sort of fell into. And then we were able to characterize a population of monocytes that expressed some markers, CD16 and CD163. CD163 is like a hemoglobin scavenger receptor that had been reported in other diseases to play both a pro and potentially anti-inflammatory role in some diseases. But we thought it was playing a pro-inflammatory role in neck for a couple of reasons. One, this population of cells were present next to the epithelium in neck samples. We looked at that kind of using spatial imaging mass cytometry. And then using some single cell RNA-seq, we were able to show that they had a pro-inflammatory signature. That was kind of a crash course in translational research, going from how we get the samples to identifying appropriate controls for an experiment, to sort of analyzing the data that we got, and that has kind of expanded into looking in the blood because we did see in some patients that we had some blood samples for this population of monocytes, and then also looking at spontaneous intestinal perforation because that had a different signature from neck.
And for those SIPs, that's the focus of your K award, right? I think.
Yeah, yes. Yeah, so that right. And so when I, kind of saw that, oh, like we know even less about spontaneous intestinal perforation, and we can see this sort of signature that's suggesting that there are some immunologic differences, at least at the time of injury in SIB, really suggesting that there might be some inflammation. You know, till now, what we know of SIP is that like a small percentage of preterm infants get it. Maybe it's associated with some hypoxia. Maybe it's associated with some placental abnormalities. There's some literature about relationship between endomethacin and postnatal early postnatal steroids, but like it's really poorly understood. And so seeing some changes within the immune landscape of the intestine potentially suggests like there's something else that might be going on, and that sort of really inspired me to want to look more um, more closely at it.
That's great. It's so important because, like, a lot of the early neck literature was contaminated, I think, a lot by spontaneous intestinal perfs and, not being able to differentiate because they didn't have an exploratory laparotomy, not being able to differentiate, was it neck or zip? Um, I think that's one of the major weaknesses of our field, obviously, but as
I'm curious if you could speak to this idea that are, I think you're one of the people who's most able to speak to, are SIP and neck on a continuum? Are they totally different pathophysiological processes? We were having a discussion about this in M&M the other week, and I'm looking forward to seeing even more. Like congratulations on a number of remarkable papers over the past couple of years. But I'm excited to see the SIP
Yeah,
papers come out too.
yeah.
Come on, Boomy. Are you busy? Yeah.
Yeah, yeah. I mean, I've been busy sort of trying to answer that question. And I think like Misty kind of alluded to, one of the challenges is kind of in there is a slight overlap in presentation, right? Like typically we think about SIB as occurring within the first 7 days or within the first 10 days of life, but there are a subset of patients that could develop um, like a neck. They might have a perforation, but it might end up being sort of neck pathology. And so on a pathological level, they are sort of distinct, right? Usually SIP is associated with, we know NAC is associated with transmural necrosis that can be sort of like diffuse. But SIP sort of histopathologically has been a little bit more vague. And so sort of historically, one thing that had been described in the literature is that it's associated with some deficiency in the musculars, um, in the muscle layer within the intestine, but that's only seen in about a third of patients. And sort of like more recently, right, like the pathology might describe sort of like a focal injury without sort of like villus villus blunting. And so far, like the work that we've been able to see, at least like looking specifically within the immune landscape, is that in patients who develop SIP, there is a distinct signature in the T cells um, compared to the signature of T cells in neck. Specifically, these T cells in SIP have a pro-inflammatory signature that's characterized by an increased expression of interferon gamma. And then they seem to have lower expression of some chemokine receptors, CXCR, namely CXCR3, which I had initially thought that that meant that like maybe there's some failure of like migration of the T cells into the intestine that's increasing susceptibility to SIP. But we're still sort of, in the process of doing some more studies to further understand, why there is this signature and if it's just that SIP is affecting just the T cells or if it's sort of like more kind of global epithelial and immune cell damage. To answer your question about do they exist on a spectrum, I think that that would be like my most common hypothesis is that, there's sort of like in my mind, kind of like inflammatory bowel diseases of the neonate, sort of like how we, conceptually think about IBD as being like Crohn's versus UC. SIP is this sort of disease that um, has is typically localized to the terminal ileum. Meanwhile, neck can really sort of affect um, anywhere in the small or large intestine.
Yeah, thank you. I'm really enjoying this because it's all of the questions that I don't know, that I think about. Why early? Why is one contained and the other
Right.
not? Do you think, um to ask a very, very simplified question as
Yeah.
just like a way to, get you to talk about other exciting things. Like, is it the difference in a T cell population being affected versus neutrophils? I mean, I also, from a little bit of work we've done on germinal matrix hemorrhage, think of neutrophils as bad actors in general. And T cell populations are a little more refined. Sometimes they're good, sometimes they're bad.
Yeah. I think that that's fair. I think that like it's likely a little bit more than just the T cells. Um, I think that potentially you know one one thought that we've been sort of exploring is that as we learn more about how the mucosal immune landscape develops in humans, both in utero and in early postnatal life, um, we know that there's going to be sort of like there are region specific differences in how the T cells and other cells develop. For example, like we know that as early as 12 weeks in gestation, the T cells within the intestine are predominantly memory T cells. And so they have this sort of like functional phenotype. And that sort of,
Wait, can we nerd out there? How do you have a memory T cell in a sterile environment in
right.
utero?
And so this is the other sort of like big debate is like, is it in utero environment
What?
complete?
Wild! Yeah, okay. Tell me
I don't know.
more.
Is the immune is right, is it
Is
is
it sterile?
is it completely sterile or is it are these memory T cells sort of responding to um, you know, metabolites, bacterial metabolites that are potentially within the um the amniotic fluid? Because like, amniotic fluid swallowing kind of begins early on. And the way that the intestine development would occur would be from sort of like proximal to distal, like everything else. And so yeah, so lots of questions, lots of things, lots of things to learn. But I think that the understanding of fetal mucosal immunity has sort of like expanded significantly in the past 5 years, and that sort of is informing how we think about both of these diseases, and it's kind of informing sort of our approaches um, to studying them, and I mean, I think that, the biggest pros are, kind of access to, um the biobank of samples that we have and sort of collection is always ongoing. And also sort of the explosion of and the development of like various single cell
Mhm.
approaches that allows us to really get a lot of information
Mhm.
from small tissue samples.
You had mentioned earlier, you'd mentioned about, you know, in the medicine, postnatal steroids. I'm curious, and I'm sure our listeners are curious too, like if you've been able to identify any other medications that predispose an infant to neck or any other, I don't know, practices that we do every day in the NICU that could be predisposing our babies or setting them up.
Yeah, I mean, I think that that's sort of a great question. As we, in terms of neck, as we all sort of are know that like or we think about how human milk is protective. But I think outside of that, our understanding of the pathogenesis and how various factors potentially contribute to the disease is so ongoing. I mean, so far in our work, when we um, looked at a cohort of preterm infants, they were less born at less than 30 weeks gestation, and we sort of had collected longitudinal blood samples um, to sort of understand how the immune system changes over time and how that's modulated by various postnatal and antenatal exposures. The only sort of link that came up from the small cohort of like 10 patients was this link that we sort of already know to like histologic chorioamnionitis. We saw that that specific exposure sort of modifies the population or alters the population of monocytes. And that change is persistent even at sort of 1 month of life in these samples because we looked at blood in the first week, first month, and at 2 months. And so I think it's hard to say if there are any other specific medications that change that. Certainly we think about how antibiotics could be potentially um, detrimental to the microbiome, but we haven't. Yeah, we're still sort of still looking. These questions are tricky because um, neck, as we know, is sort of heterogeneous disease, and it happens, but sometimes not that often, which is good for the babies. And even when it happens, there's always sometimes a lot of confounding factors. So
Yeah, it's
yeah,
definitely 2 hard diseases to study, right? Neck and
yeah,
and I think, for anyone out there that's thinking about doing translational research, like collaboration is important, right? To be able to, get the samples and be able to analyze them. Like it's really, really team science and all of this is a team sport. So we're all anxiously waiting for you to tell us the answers. Um, um, you have your whole career cut out for you to figure it all. But but yeah, no, it's nice to hear about your K and studies related to that. So great.
Can I nerd out for a little bit longer?
Yeah, yeah. Go ahead.
I can feel Misty's getting ready to talk, like, about the big picture, and I'm like,
yeah, yeah.
"Ah."
Well, now I'm wondering if you're gonna ask if we thought about sort of like how the vasculature play a role since you're a vascular biologist.
No, that was question number 3. There was a question 1 and 2 before that. Oh, I feel seen. Thank you. That's awesome. Oh, thank you. Okay, but the back to sip and neck. The question I wanted to ask is about. I mean, I think it does really speak to the um see these precious, precious human samples. And I mean, I think it is wonderful that initially that you didn't have fear when you were handling them, right? Because I that's not good either. You see the person who's like shaking when they do the experiment. And I have mixed feelings because on the one hand, it is recognizing that there was a human being that contributed, to the material that you're currently holding. And I don't know, sometimes it's helpful just to not think about that and just, like, pipette. Um, so the question that I wanted to ask was, animal models, do you feel like there's a future where we can model this in animal models? Or, um, have you I don't know, how do you feel about Misty's neonatal mouse model? Or are you thinking about trying something different with, I mean, a pig, a monkey. Yeah, I think that, there's some exciting development in translational models. Like I'm collaborating on a perinatal choreo macaque project right now because I think, there is a lot of interest in trying to be thoughtful about what models we're using to answer different human specific questions.
Yeah, I mean, I think that that's a great question. And I think about that and how to sort of transition from where I am, right, sort of like mid K to to like R and next steps. I
Good. Yeah.
right now I don't have a great answer. I'm still sort of like it's still at the drawing board. But certainly, I mean, I think that like potentially if anyone had some ideas and was interested in collaborating, that's sort of the route to go. Um, I think that what we've been trying to work on is potentially using organoid models. And
Mhm.
so that's kind of the stage that we are where we're at. So there are obviously challenges with that as well.
Yeah, I think I was just in terms of animals, like, modeling SIP in a neonatal mouse, is it's, it's not doable. It's definitely doable, but the I mean, it's not, there's no good spontaneous intestinal perf. There's more or less, an intestinal perf type model that, you could certainly do with, human enteric microbiome, IP injected, into a neonatal mouse, which has been done and, like, perfected by Jim Wynn. He did an amazing job looking at, he was using the model as, like, a neonatal sepsis model, and, um, and so you could certainly do something like that. The spontaneous aspect of that,
Yeah.
it's not well understood, right? What drives intestine to get a gut barrier breach, really, right? So, uh,
Right. And I think that like that and that's sort of like the key question. And I think that's kind of like the limiting factor, at least from where SIP is concerned about sort of trying to replicate this, you know, the spontaneity
yes.
of it.
Yeah, but at least with your data set, you could sort of take a Misty Good approach, which is like take all of the things that lead to neck, put them together in one poor neonatal mouse, and then, sometimes you get neck, right? So, and neck in an animal model, and so I think it'll be exciting to see all the things that come out from, your very sophisticated immune profiling in that disease, which is where I think where it needs to start. Um, yeah, I guess another question was just which in terms of the different neutrophil populations in neck, which one do you think is which one do you think is affected by breast milk? Or I don't know, do you have other thoughts about their heterogeneity in terms of the pathogenesis or the ability to, flip it the different way, which immune population maybe is involved with some resilience?
Yeah, yeah. I mean, I think that when I think about sort of like breast milk and, immune populations, at least like what we've what's like currently known is sort of how lactoferrin from breast milk modulates the MDSCs or myeloid derived suppressive cells. And so I think that that's the population, and that we know that in patients who have neck, this population is um, sort of affected, and but exposure to human milk kind of um, helps modulate that response. Um, I think the other piece from sort of like breast milk and neck protection is also sort of out of, I think he was Pittsburgh, out of Tim Hand's lab. They had been looking at sort of the impact of like maternal IgA on sort of from breast milk on on neck, and they showed that maternal IgA binding a specific bacteria could be sort of protective for neck in some patients. Um, and so I think that ultimately there are likely, multiple components of breast milk that are modifying the immune system. The other sort of modifier that we know is like human milk derived oligosaccharides, and how breast milk contains these molecules that don't get digested but are basically like food for certain types of bacteria to promote the development of certain bacterial colonies like bifidobacteria, for example, and that piece is protective for neck. So like I think that we know kind of all these um, these pieces, and so that's kind of interesting, as well.
Yeah, thank you so much for that overview. I think this is something that when I'm, rounding with the residents and I want to say like what do we know in terms of like the molecular pathogenesis or resilience against NEC, I will refer them to your overview. So thank you. I think that was a nice educational piece too. Yeah, I mean, I won't hesitate to ask the vascular question. If you have anything you wanna if you have, any other brilliant insights,
Well,
but we don't have to go there either. I think we've had a wonderful discussion.
I mean, yeah, I think that we do have some like in one of the sort of like unbiased looking at neck using sort of single cell data, we did see that there are some there's like again sort of pro-inflammatory signature in some lymphatic endothelial cells in patients who have neck compared to those um, who do not have neck. And so I think that likely there's some, and as I mentioned earlier, the population of CD16 positive, CD163 positive monocytes that we saw um, spatially were in the intestine, but they were adjacent to blood vessels. And so there's likely, and as if we think about how some studies have shown a drop in peripheral monocyte count right before the diagnosis of neck, we can say potentially hypothesize that these monocytes are being recruited into the periphery through the endothelial cells, and are either sort of responding to injury, or are they the ones driving the injury? That sort of
Mhm.
question is unanswered. But I do think that, you know, the endothelial cells play play a role um in either the development, or more likely the sort of propagation of the inflammatory cascade.
Yeah, I think that's so interesting. Like, not a lot is known about the lymphatics and the association of neck or, what what's going on. So hopefully, you and now that
Yeah.
can certainly share some insights there. But
But if you want to, as a vascular biologist, you want to weigh in. I mean, but then I sort
I
of think
mean,
about
should we start sketching out the aims for our collaboration
we
now? I
just
mean,
should do it now. N-
just,
n-
to sort of think on the spot and show folks how and maybe we could, submit it. I
Yeah,
mean, yeah, please.
yeah, no, no. I mean, I just sort of then think about how, patients who develop neck, sort of have this kind of, um, white matter injury in either
Mm.
in response to the inflammation. And I know that David Hacken's group had really nicely shown sort of how the CD4 T cell population was sort of like pro-inflammatory and sort of migrating to the brain with some really cool mouse brain organoids. But anyway, there's clearly like a gut brain axis. And and then the cellular cells are most likely involved.
Yeah. Yeah, I mean, I think these are all just, um, I don't know, really, enticing, like, observations, right? Like, scientifically and clinically from the samples and the molecular profiling that we're all getting a better handle on now. So, yeah, I mean, Brisa Pelacucci, do you know Brisa, actually? She's at
Yes,
Yale
yes, yes.
now.
Yeah, yeah. Yeah, she's very interested in sort of endothelial cells and the regional specialization.
Mhm.
We're connected, so
That's
that's
awesome.
that's
Yeah, I
cool.
mean, she had probably the first characterization of the human intestinal lymphatic vasculature, so that's an awesome local person, but yeah, I mean, I think there's this outdated notion that the vasculature in terms of immune cell infiltration is like a door that's either open or shut, and I think, I mean, the paradigm I think about the most is the blood-brain barrier in regards to that paradigm. And finally, we're adopting a more nuanced understanding of, like, it's not open and closed doors. It's probably like, many, many stages of gating in terms of, like, as you alluded to also, like, the T cell needs to adhere to the endothelium. It needs to, diapedese across, then get across to the ECM, and all of these are kind of um, chemokine related events also. So, when is that potentially physiologic and when is
Mhm.
that um, pathologic? Those, I mean, the good news is they're discoverable things. We
Right, yeah.
just, need to keep like doing the work that you're doing. Actually, all of us are doing
Right, yeah.
um, in terms of taking the human samples, understanding the molecular players, and then really probing them kind of in a direct functional way, and hopefully we do have, I guess, I guess, could you imagine for us a future in which, like, we have something that prevents against SIP, or we have a therapy against it once it
Yeah,
happens?
I mean, I think that that would be great. For SIP, I sort of imagine, like, maybe something given to sort of, like, high moms that are at higher risk, just given how sort of early the incidence is. Um,
Mhm. Yeah.
but
And
potentially.
I was gonna zoom out a little bit now, Betsy.
Please. Please, yes, and as we hit the 45-minute mark, so thank you, Misty.
No, I'm just curious because so many of our listeners, are physician scientists and they really are probably wanting to know because you mentioned your mid K. And so thinking about the K to R transition, which is, as
Yes.
you know, this like very vulnerable place where people sit. And I have sat there and, you know, had fear. And you had mentioned fear initially, like that you didn't have any fear to go into the lab. But I think the K to R transition is one that a lot of people have fear. And I'm just wondering if maybe you could give some advice on like how you're thinking about it for your own career and then how to help overcome that fear.
Yeah, no, I'm terrified, of the Katie Couric position. No, as I mean, I think I've sort of approached, my career so far and like, we're just gonna take it 1 year at a time, and so I have a plan for this year, and, that includes, you know, taking the local grant writing course again and,
Mhm.
sort of really tapping into the resources, like mentorship and getting feedback early. Um, and and then yeah, the grant writing course here was super helpful when I first submitted my K and there were people writing ours then. And so it's just sort of getting as much feedback in the writing stage as possible. Um, I hope is helpful. And then, yeah, again, like you submit and it doesn't go well the first time, be willing to and have plans to resubmit. Is sort of the approach. Understanding that, like, I guess one of the benefits of being a physician scientist is like yes, there is pressure, to get that R, but I also still enjoy taking care of babies in the NICU. And so
That was literally how I got through. I remember thinking, "Okay, well, if it doesn't go well, like, I'll just take care of babies and be totally happy," you know?
yeah,
Uh, I think that's one of the things that when you're in it, you tend to lose sight, and it's I don't know, I felt like if I put less pressure on myself to get the R or transition, you mentioned transition to independence too. I think that's one of the for me, it was one of the biggest fears is the transition to independence and running my own lab and
yeah,
all of the things that go along with that because it's fun to do science. And so when you're trying, you're doing the experiment or doing the analysis, but then, taking that next big step where you're leading a team and
yeah.
inspiring the next generation and mentoring, all of the fun things that is really cool about being a PI.
Yeah, thank you. We like to end on something fun about your research, or about your teammates in the lab, or about your colleagues in neonatology. Um, yeah, something. Tell us something that you enjoy, to do as a team, or just something you personally enjoy when you're not pipetting, or looking at the AEG screen overnight like I was.
Yeah, yeah. I mean, I think when I'm not here or and before I became a mom, I always sort of looked forward to playing tennis, and a lot of the folks in the lab play tennis. It's all indoor tennis because we're in New England, but that's something I'm hoping to sort of get back into. And now, now that I have a little 10-month-old at home, I basically just look forward to seeing her at the end of the day and hope she sleeps through the night.
Yeah, and you run after her, who's essentially, well, I guess not quite yet. Not yet,
Yeah, not
like,
quite yet. Not
not
quite yet.
yet
She's she's
ping-ponging.
she's she's
Yeah,
scooching. Yeah, I can't wait.
yeah.
I don't know.
Oh, how
I can
cute.
wait for the day she starts running around.
Yeah,
Yeah.
exactly.
And then any, like, pearls of advice or wisdom or anything that for all of our early stage investigators listening?
Yeah, I mean, I think that sort of like the biggest um and I think this has come up in a few other episodes, is just sort of finding, good mentors who are sort of thinking about, you know, how your work fits into their work right now, but also thinking about, what's next, and being um very sort of helpful in connecting you with various collaboration sort of locally and internationally. Um, I think a few times I've mentioned Lisa Konnikova, but she's been a great mentor. Like, she let me join a lab when I didn't know anything about immunology. And the lab had moved during the pandemic to Yale, and I got an opportunity to come here to Yale, and as a postdoc and an attending right out of fellowship. And sort of these things have been incredibly instrumental in helping to um, move my career forward. And so I think that finding great mentors and just being sort of willing to learn and showing up even when you're like, I have no idea what's happening.
Well, I think that's great, and not having any fear, right? That, like, you can't do it because clearly we've all shown on this podcast, like, that you can do it and you've been incredibly successful. And I think for you, this is just the beginning. So keep going and you're really asking the questions that we all want to know at the bedside,
Mhm.
you know? So it's worth it. It's worth it. for the babies.
Awesome. Yeah. Thank you so much for those kind words and for having me.
Yeah. Thank you for coming. We are just really thrilled to host you and highlight your work and are grateful for all the work that you do.
Yeah. Thank you again, Bunmi, for joining us and for enlightening us, and to our listeners for engaging in our conversations as well. Well, that's it for today's episode of At the Bench, and we look forward to talking with you all soon. Take care.